Okay folks, I think we'll get started. It's a great pleasure to introduce Fraser to you all today. I think most of you know him. Fraser originally heralds from Scotland. He did his undergraduate and master's degrees at Newcastle University, where I first met him. He then went on to work for the Wildlife Conservation Society in Papua New Guinea for several years. And Dave Feary, Josh and I went up there and collaborated with WCS, including Fraser, in 2009. And that's when we first started looking at this idea of fish flight behaviour. And it was clearly a topic that needed a lot of research and was the impetus for Fraser to get stuck into it for a PhD. Now there's a couple of things that I want to mention about Fraser's PhD. One is he spent the last year or so living in America because his wife, who did her PhD here in the centre, got a job over in Madison. And that's not an easy thing to do, and a lot of people slow down or even don't finish when they go overseas before they've finished their degree. And Fraser's done a great job of keeping up that momentum, publishing, and keeping on track with his timelines. So, that's a great thing that he's managed to achieve. And secondly, this topic really was wide open at the beginning, so it hadn't really been looked at in coral reef fish at all. And so I think Fraser's done a really nice job of, first of all, looking at a lot of the methodological biases that might exist in this kind of work and putting those to bed and getting the methods and the techniques standardised before going on to do some really innovative and important applied research that's relevant to the management of coral reefs. So I think it's a really nice way that it's built the thesis together. So with that, over to you, Fraser.
Thanks, Nick. So today I'm here to speak with my pre-completion seminar, The Fear of Fishes, Anti-Predator Behaviour of Coral Reef Fishes, and it's relevant to fisheries management and conservation. So before I get into the meat of my talk, I'm just going to quickly go through the outline of it. I'll talk a little bit about the background behind the behaviour I'm looking at, which is primarily prey escape behaviour. I'll talk about how it's been used in the terrestrial realm and go a little bit into the theory behind it. And then I'll try and bring through why this behaviour is relevant to coral reef fisheries and identify some of the gaps in our knowledge. I'll address these gaps through my aims and research questions, and these are basically put into two broad scenes. The first of them is how human predation influences escape behaviour, and the second of them is what this actually means for management. I'll then go on and just give some of the results from my research questions, then move on to some conclusions and future directions that I think this work can go in, and then progress, publications and acknowledgements. So first of all, looking at the background, why behaviour? Well, as Nick has mentioned, behaviour is generally understudied in coral reef fisheries. I'm particularly talking about coral reef fisheries, not coral reef fish. And this is a bit strange because we know that often behaviours are exploited by fishes, particularly behaviours sort of like on a large scale at the school or the species sort of level. So spawning aggregations, where fishes come together in very large densities, are often targeted by fishermen, primarily because they're predictable and you can get a very high catch-free net effort, a very high level of fishing efficiency there. We also know that fishermen, particularly in Melanesia and the Pacific, will often target where fish rest at night. So Boldometon miracatum in Palau, for example, and in many other places in Melanesia, are often targeted by fishermen at night because they know that they come into these shallow lagoons, they know where they rest, and again, very high catch-free net effort. The other reason is that gear efficiency on coral reefs is very likely to be impacted by behavioural changes. A lot of the gears being used, particularly in Melanesia, are relatively low-level technology, and they can actually be and whether or not they're successful can really be determined by behaviour at the individual level of the fish. And finally, in much of Melanesia, traditional management, specifically in the form of taboos, but more and more now in locally managed marine areas, have been used specifically to alter behaviour to make fishes more catchable. And I'll talk about this a little bit later on in my introduction as well. So moving on, the tool I'm using primarily to look at prey escape behaviour is flight initiation distance. And this basically is how close a predator can get to a prey animal before the prey animal flees. This is heavily used in the terrestrial realm. There's a 50-year history of this being used. And generally it's thought that under conditions of high predation pressure, prey will flee earlier and then encounter with the predator. As I said, this has got a fairly long history in the terrestrial realm. The human impact on behaviour and flight initiation distance, I'll call it FID because otherwise my maths will dry out halfway through. So it's been used to look at habituation. Specifically, it's been looked at habituation of animals which occur in urban landscapes and see how this influences their behaviour and various things like breeding success. And more now it's being used to look at the effects of hunting on animal behaviour. So it's been looked at illegal hunting and poaching through sort of like these formalized management areas, and also through these very sort of like these things which have a long history of being exploited and a long history of sort of managed hunting, things like capercaillie and deer. Just diving down a little bit more into the theory behind this and why it's important. The theory underpinning flight initiation distance. So first of all, the theory that sort of dominated at first was this perceptual limit hypothesis. And this basically said that flight occurs when a predator is detected. So this really got into aspects of group size, the amount of time that an animal would budget towards feeding versus scanning for predators and so on. But it was found that this really didn't explain very well a lot of the patterns that were being detected. And so in probably the seminal work in the field by Udenberg and Dill in 1986, they formally stated an economic hypothesis of fleeing from predators, which was that flight occurs when the cost of remaining exceeds the benefits that you can gain by remaining. So a little bit more detail on this. The cost of flight is generally considered the energetic cost, so basically how much it actually costs you to run away, versus the opportunity cost. And the opportunity cost is considered to be by far and away dominant in the terms of the cost. It's basically energetic cost is considered almost negligible in the theory. And so this means that as the distance from predator increases, the cost of flight will increase because you're foregoing more benefits that you could otherwise have obtained. The cost of remaining is generally considered proportional to the probability of capture, so at distances it's relatively low, and then once you get to a certain distance from the predator, it starts to increase at almost certainly at a non-linear rate until it gets to infinity when the distance from predator is zero, pretty much because it's assumed that the prey animal has been caught and is dead. So that's an infinite cost effectively to the prey. Now, the cost of the optimal flight initiation distance then will occur when the cost of remaining becomes greater than the cost of flight. So this is here indicated by the D star. So what happens when you increase the cost of flight? For example, if it's a rich food source which you'd otherwise have to forego, well, the sort of like the gradient of the line as the cost of flight increases, and what you find is that you have a decrease in the optimal flight initiation distance. So animals will basically allow a higher probability of capture in order to take more benefits from whatever is there. Now, the corollary of this is that if there's an increased probability of capture, which could occur through predator characteristics, the predator is particularly big or particularly fast, or the prey life history stage, for example, if a prey animal is pregnant or whether it's molting and so on, you'll have an increased cost of remaining, an increased probability of capture, and then you'll see an increase in optimal flight initiation distance. So moving on to talk about the things that will influence flight initiation FID. First of all, this was a big meta-analysis done by Stankovic and Blumstein in 2005, and this is primarily looking again at terrestrial animals. There's a few sort of like stream fishes from gobies and so on in there, and guppies. And they basically found various external factors to be important, things like predator speed, size, directness of attack, and resource quality, with some predator characteristics being far more important than resource quality. They also identify some intrinsic factors, and the three that I'm really going to concentrate on here are experience of predators, basically whether or not you've either survived or seen a predation event, the size of the age of the individual. So the theory behind this is that if you're a particularly large individual, the relative benefits of remaining are going to be much lower than if you're a small individual. So a large individual will choose to flee earlier, and also group size for various things like predator scanning, predator detection, and so on. So, what relevance does all this have for coral reef fisheries? Well, humans are a major source of predation on coral reefs, and arguably the major source of predation once fishes have exceeded a certain size. And there's a lot of anecdotal evidence of deliberate alterations of escape behaviour for EDA fishing. As Nick alluded to, one of the main things that set all this off was a conversation that Josh had in Karkar in 2001 with the village chief, where he basically stated that the reason that they're closing these areas for 6 months to sort of like 2 years is specifically so that they can get closer to the fish and catch them. And in 2009, as Nick said, we went and did some of this work. This was work led by Dave Feary. And here we have 7 species of fish. We have 5 primary target species, and these are species which are particularly targeted by spearfishes, and 2 secondary targeted species which tend to be more targeted by lionfishes but still occur in the spear gun catch. And the dark bars are in the unfished areas, the light bars are the fished areas. And what we saw was that there was a significant difference in FID between the unfished and the fished areas, and that this red line represents the mean effective range of spear guns in the area. And quite often you see that outside of the protected area, so the unfished area, FID exceeded the mean effective range of spear guns. So this leads to another sort of question. If FID profoundly affects catchability, then we have this issue that catch per unit effort may not reflect actual abundance. You may still be having quite a high catch per unit effort at a relatively low abundance. So you have this sort of mismatch, and this might have various implications for fisheries management. We also saw, what about permanent no-take reserves? So we've already seen that there's this sort of increase in catchability, this decrease in flight initiation distance in sort of like temporary protected areas. But permanent no-take reserves, we'd also expect to see this inside permanent no-take reserves. And we know that we have biomass build-up and spillover through density-dependent effects in no-take reserves. Why this might not also happen for behaviour is one of the questions that we wanted to get at and look at. And finally, as I mentioned before, this is well studied in the terrestrial realm, but we have major questions remaining whether or not lessons from the terrestrial system are really applicable in the marine environment. First of all, the medium that we're looking at, water versus the terrestrial realm, is very different, and there's a whole host of different sensory cues which might be around. So whether or not some of the things that people have said are actually important is something that we need to look at. So this leads me into my aim and research questions. So my general aim was to explore the role of escape behaviour in coral reef fisheries. And as I said, I did this through two main themes. The first theme was the impact of human predation on escape behaviour. And I looked at two questions that fall into the theme. The first is what factors influence risk assessment and the escape behaviour of coral reef fisheries. And the second one is there a consistent relationship between fishing pressure and escape behaviour? I then moved on and looked at the implications for conservation and management. So my third question was how do changes in fish escape behaviour influence benefits obtained from periodically harvested closures? And my final question, getting at the spillover, does reduced FID extend beyond the boundaries of no-take marine reserves? Okay. So my first question was about what factors influence fish escape behaviour. I basically have replaced a spear fisherman for the leopard and a fish for the springbok, I'll get that wrong. And this is basically what I'm doing. I'm using a spear fisherman or a proxy for a spear fisherman, basically to try and get flight to occur and measuring flight initiation distance. And at first I wanted to look at the sort of external factors, predator speed, predator size, and directness of attack in particular. And I wanted to control for these so that I could do consistent work throughout my PhD. And so in order to get at this, I shadowed spearfishers in Papua New Guinea, and I looked at what they did, and I tried to remove any idiosyncrasies from their particular methods of spearing fish, and tried to get at what everybody had in common. And so I created this standardized technique where I would descend from the surface about 8 to 10 meters away from a target fish. I'd then sort of like lie along the substrate, sort of parallel with the substrate, and at the same depth as the fish for about 20 seconds or so, just to make sure that the fish hadn't fled or it wasn't disturbed by my descent. Then I'd approach the fish at a steady speed and dropping a marker at where my head was when the fish fled and from where underneath the fish was when it fled. And the measuring the distance between those was the FID. I had to define flight properly to this. So one of the big advantages in a terrestrial system with a lot of the animals that they use is they're looking at things like roosting birds or basking lizards and things like that, which don't move, whereas fish move around quite a lot. So I had to define when flight occurred. And so for the purpose of this research, I said flight occurs basically when a fish it starts to move away from you faster than you move towards it. So not just when it's just generally moving around the reef, but when it actually increases speed and starts to move away from you. I also controlled for interfish interactions. Basically, if a fish gets chased by another fish, chances are it's not responding to you anymore, it's responding to the fish. And I obviously didn't want that. So any time that did happen, I sort of abandoned my attempt at estimating FID for that particular fish. So moving on a bit, looking at the methodology of what I was doing. So again, I did this work in an MPA in a fished area, so I wanted to get this management effect, which I knew occurred, and wanted to see if there were any interactions between various different methods I used, and any biases that might come out through this. I asked, does the use of scuba result in a lower FID than freediving? Most of the work, there's been 2 studies done on this before my PhD, and both of them were done in scuba. However, scuba doesn't most spearfishermen don't use scuba. They tend to free dive, and so if we're looking at trying to be a predator, sort of like a predator in these regions, we really need to be a free diver rather than a scuba diver. Do fishes respond more warily if the diver has a spear gun? In many of these areas, I couldn't go into the marine protected areas with a spear gun. It just wouldn't be culturally acceptable, or even a spear gun substitute. So I needed to find out whether or not fishes respond more warily if a diver has a spear gun or not, and whether there's a bias associated with this. And finally, I wanted to know if there's a difference between observers. One of the things I really want to look at is whether or not there are variations in how people approach. And so we had 3 different observers who were looking in each of these treatments. So this gave me a total of 24 treatments, so 3 observers in 3 observers in 2 gear treatments, which were nested in 2 dive treatments, which were nested in the 2 protected management treatments. I did this, and each observer basically did trials on 15 Scaridae in each one of the treatments. What were the results from this? Well, first of all, is there a difference between observers? So this graph is the difference in FID between observers. The error bars are the 95% confidence intervals, and so on the left you have observer 1 minus observer 2, middle 1 minus 3, and final 1, 2 minus 3, and as you can see there is no difference between observers, which was quite a nice little result, because it means that this is probably going to be a replicable technique across observers and across different places. Do fishes respond more warily if a diver has a spear gun? The analysis I did for this was actually a ANCOVA with fish size and group size included in it as covariates. So whilst these 95% confidence intervals, again, this is difference in FID between treatments, don't overlap 0, there wasn't actually any significant effect here. And so, no, we can say that there is no real difference between whether a diver has or does not have a spear gun. Does the use of scuba result in lower FID than free diving? Again, we saw no effect here. No difference between treatments. And finally, is there a management effect? And the answer is yes, there's a very large management effect, and this probably accounts for about 60% of the variation in FID. So these are some of the biases that I've identified to date, and so I could take some of these lessons and apply them throughout my PhD. Moving on to my second chapter, I did this work in New Ireland province in 4 reefs. The reason we went up to New Ireland province is I'd worked at WCS, Josh had done some work there, and we knew that we could have a gradient of fishing pressure. We had details about what the fishing pressure was in various villages and on various reefs. And here I was looking at these sort of intrinsic factors, the experience of predators, so that gets at fishing pressure, so the size of the age of individuals and the group size. My general hypotheses were that FID will be larger in bigger fishes, that FID will be larger if fishes are in larger groups, so this is going along the terrestrial sort of predictions. And then finally, that FID will increase with fishing pressure. Basically, as you get greater experience of predators, you'll see an increase in FID. The methods I did the FID surveys that I outlined at 4 reefs with different fishing pressures in New Ireland province, taking note of things like fish size, group size, and so on. And I also had fishing pressure data from previous household interviews that Josh and WCS had conducted. Going on to my results, this is a graph of flight initiation distance against the total length of fish for just 2 families, Acanthuridae and Scaridae. And in general, we found that larger fishes have a greater FID, and there's a significant relationship here. But we did find that this varied a little bit. It varied by family. So, as I said, this is Acanthuridae and Scaridae, where we had very strong relationships between total length and FID for across at least 3 of the 4 reefs. Whereas for things like Tyranidae and Legenidae, we only had a link between FID and size for a couple of reefs, and it was a very weak relationship. We also found that it varied by predation pressure, by fishing pressure. So each of the different colours of the symbols here is a different reef, and generally we found that as we increased predation pressure, there was more of an effect of size on FID. There was little effect of group size on FID. In fact, it only occurred for 1 family at 1 reef, and I was looking at 6 families in this particular analysis. And so group size really didn't seem to be very important in the marine realm. Because of this result, I've basically accounted for size now in all of my future analysis, and size normally using it as a covariate, sometimes actually using it to weight models and so on. So moving on, does fishing pressure influence FID? Well, here for the Acanthuridae and Scaridae, which are again primary target species, so these are families in this area which are heavily targeted by spearfishermen. What we saw was that as the gradient of fishing pressure increases from 0, which is the lightest red bar, to high, which is the dark red bar, we did see an increase in FID, but it did differ a bit between the families. The black lines above the bars are basically similar groups. As you can see for Acanthuridae, we only really had the increase at the highest fishing pressure, whereas for Scaridae there was more of a gradient of FID as fishing pressure increased. Moving on, the secondary target families I was looking at here were Mollidae, Ballistidae, Serranidae, and Legenidae. We found similar sort of results for Mollidae and Ballistidae, but Serranidae and Legenidae, which tend to be the ones that are most targeted by lionfishes, and Legenidae tend to actually stay deeper and generally out of flight, sort of like mean effective range of spear gun more often than other families of fish. We found no sort of like similar result of FID increasing with fishing pressure. So, as a result of this, for the rest of my studies, I actually concentrated on Acanthuridae and Scaridae, primarily because I knew that they were being heavily fished across every single area I was looking at, and also because of the numerical abundance on reefs. Basically, in order to get around and sort of account for sort of like statistical independence and power analysis and so on of my work, I wanted to make sure that I could actually observe FID for enough individuals on each reef. So Acanthuridae and Scaridae were chosen for my families for the rest of my work. This is my chapter which I'm currently working at, so the analysis for this is only in preliminary stages. And I wanted to expand what I saw from this fishing pressure gradient from a fairly small area across the broader Indo-Pacific. And so again, this is why I wanted to look at the methods at first to make sure I had a standardized technique, so I could actually compare areas across the entire Indo-Pacific. And so the work I did in PNG, here I've also got some data from Vanuatu, through my PhD, from the Philippines, and then Nick collected some data from Chagos in early last year that I could use in this analysis. What the results are? Well, this is flight initiation distance again against fishing pressure in fishes per kilometre of fringing reef, so that's per kilometre of reef front. What we found was that there's a significant increase in FID with fishing pressure, very consistent with the results we found from PNG. I'll say this is just for the Acanthuridae at the moment. I haven't gone in and looked at the Scarids yet. But the really interesting result that we found was that for protected areas, so the blacks are unprotected areas, and the sort of blue triangle are protected areas, we also found an increase in FID with fishing pressure. And so we found that protected areas embedded in fish landscapes still show an influence of fishing on escape behaviour. This particular graph is adjusted for sort of like country effects, and so the blue triangle on the left-hand side is Chagos. And when you don't adjust for country effects and the fact that Chagos is in this completely unfished population, the Chagos FID is very, very low relative to everywhere else. So this is a really interesting finding. I'm quite interested in looking into what else I can find from the data to sort of explain this relationship. So this has me answered some of the questions about what factors influence FID. And so I'm moving on to the management aspect. My third question was, are there benefits from fish behaviour for periodic harvests? So periodic harvests and periodically harvested closures are a very common method used to limit access to marine resources across the South Pacific, and they tend to have social underpinnings, not conservation. So because they've got these social underpinnings, because breaking a periodically harvested closure to the area can come with quite a lot of social effects, they tend to show higher compliance in periodically closed areas. They tend to be used as a ritual or a feast traditionally. So say a big man in the village would die, you'd close an area for 6 months, maybe a year, and then you would harvest it very heavily for a couple of days, and then you'd be able to have this big feast where you have lots of fish and to honour the man's memory. They're often used as a savings account as well, so you have an area which is set aside, and then if you have to pay compensation, for example, or for school fees, you've got an area where you can actually catch a lot of fish very quickly and generate quite a lot of money very quickly. And significantly, the studies that have come out so far have shown that they probably have a similar effect on fish biomass that has permanently closed areas. So they're proliferating throughout the Indo-Pacific, and this is possibly why, because they seem to be a very good management tool. The work I did for this was in Vanuatu, in 2 communities in the Nunapeli Marine Protected Area, And the reason I went into this in Nunapele is because we had these two communities which had very similar protected, periodically harvested closures, which were being harvested for 3 days, 6 months apart, and were harvested very close as well in terms of time. They were both being harvested just before Christmas and in June. And they'd also been going for 4 years in one case and at least 18 months in the other case. And they were in a they were situated in a larger management plan, which allowed me to do a before-after control impact pair design for this, where I had 2 no-take reserves, I had 2 periodically harvested fisheries closures, and I also had 2 open fished areas I could do this work in. And so it was a very well-controlled design. I looked at behaviour. I did behaviour work, FID estimates, both before and after the opening of the closure, and I did this in all areas. I did biomass surveys, and again I did this by underwater visual census, and I was looking specifically at Acanthuridae, Scaridae, and Catchability, so Acanthuridae and Scaridae being the fish family, while Catchability being a control family. And I also did catch surveys. So the catch surveys, basically, I tried to get as many fishing trips during regular, normal fishing activities, and then I measured every single fishing trip that went to the periodically harvested closure, taking account of the time spent in the water when they left, when they arrived, what gear they used, which, by the way, was always spear gun, and also how much they caught during their trip. So the results from this, just looking first at FID, this is for these dark bars are my results from before the periodically harvested closure was opening. What we found was that FID in both forms of management, both in the no-take reserves on the left and the periodically harvested closures, which are the centre bars, were significantly lower than the fished areas, which are the bars on the right-hand side. And what we found that there was an increase in FID after harvest, but this was only seen for the Acanthuridae. The Scaridae didn't show any significant increase in FID post-harvest. And why this might have been is because of the maximum mean effective range, of spear guns in this particular area. So these red horizontal bars are the mean effective range of spear guns plus their confidence intervals that I got from surveying various fishermen in the area. What you can see is that for the Acanthuridae, in the sort of periodically harvested closure, before the harvest, we had FID quite significantly below this mean effective range of spear guns, whereas for the Scaridae at the bottom, they were pretty much at the mean effective range already. And so this means that Acanthuridae were probably far more catchable than Scaridae. And they were more likely to increase their FID. Moving on to looking at the catch data, so this is catch per unit effort in the top graph and the mean weight of fish in the bottom graph. The dark bars are during normal fishing activities and the grey bars are during the harvest of the periodically harvested closure. Catch per unit effort was higher during harvest for both all fish combined, so every fish that came out, and also for Acanthuridae, so all fish on the left, Acanthuridae in the centre. But for Scaridae, we saw no real difference. For Acanthuridae, we also saw that there was no increase in the average size of fish in the catch. And this is significant because previous work that had been done looking at taboo areas by Josh in Papua New Guinea had shown that there was a significant increase in fish size in the catch from a taboo area. So whilst we're having this higher catch per unit effort, it's not being associated with the increased size of the fish that are being caught. Moving on and looking at the proportion of families in the catch, so here are the sort of orange bars are Acanthuridae and the green with the Scaridae. Basically, between fish and taboo areas, those two families combined took up between 65 and 70% of the abundance of fish in the catch. But there was a significant shift in the relative proportions in the catch. There was a significantly larger proportion of Acanthuridae in the catch for periodic harvested areas. And so the sort of fish catch I was surveying during normal fishing activities was this sort of, it looked pretty much like this, where you had lots of green parrotfish and a couple of Acanthuridae thrown in. Whereas from the taboo areas, we generally had catch which was dominated by nasal species, particularly nasal littoratus, the orange-spined unicorn fish in this picture, whilst there were still a few scarids and a few other species of fish that were being caught. Moving on, so another important fact is that this increase of Acanthuridae abundance in the catch was not associated with higher abundance of Acanthuridae in the periodic harvested areas. People weren't just finding more Acanthuridae and were more likely to encounter an Acanthuridae. There was no significant difference between the periodically harvested closures and the sort of normal fish areas in terms of abundance. So there's something else that's going on, and we believe that this is increased this is decreased FID and therefore increased catchability of Acanthuridae. Just going on to talk a little bit briefly about biomass, the total biomass that we saw was not different between no-take reserves and periodically harvested closures, and the total biomass is on the top graph here. So again, no-take reserves on the left, periodic harvest closures and the open areas on the right. But we found that Acanthuridae biomass was similar in the periodic harvest closure areas to the fished areas, and this was significantly different from the no-take reserves. So whilst we didn't actually find a change in biomass pre to post harvest for Acanthuridae, there's a potential that several harvests and several years down the line are actually influencing the Acanthuridae abundance, and there might actually be a significant change when you look at periodically harvested closures over an extended time period as opposed to one harvest at a time. So moving on to the sort of conclusions that can be drawn from this. So this was a fairly low intensity harvest. We only had about 9 or 10 fishermen in each area doing periodically harvested closures and only doing one trip a day. So low intensity periodic harvest can have significant effects on FID, and this effect was lost for the Acanthuridae in as little as 3 days. This effect can be eroded very quickly. We found differential impacts on fishery target species, which may have implications for biomass and the sort of like the long-term sustainability of this particular management tool. We also found an additional and tractable benefit to local fishes. Basically, the decrease in Acanthuridae FID is strongly associated with various changes in catch per unit effort. So less wary behaviour, increased catchability of fishes, and biomass removal, at least from this one harvest, was undetectable. Moving on to my fourth question, which is one looking at does low wariness or FID spill over reserve boundaries. I answered this I basically looked at this two major questions in this two sort of like sub-questions here. Does non-wary behaviour of fishes spill over from marine reserves is the first one. And the second one was how far does spillover of FID extend across the boundary of marine reserves? And how does this compare to spillover of biomass? So this work was conducted in the Philippines, where I knew I had a fairly high fishing gradient outside of the protected areas. I could find protected areas that were well complied with, and we had quite a lot of still Acanthuridae and Scouridae that were being fished. And so my setup for this was that I estimated FID and biomass across the boundaries of 3 no-take marine reserves that were very well protected, and also 3 control reserves, which were these open fished areas. So what I did in an open fished area is that I basically created a pretend boundary. I planted this pretend boundary at least sort of like 800 meters to a kilometer away from the reserve boundary, and then I basically did all my work across that pretend boundary like I was doing across the reserve boundary. And so I looked at 2 fishery target families, and I estimated FID from 200 meters inside to 200 meters outside the boundary at both the reserve and the control area. And I also looked at 1 non-fishery target family, the Catchability. So moving on to some of the results. So the different coloured dots here are just the different control areas that I use, the different fished control areas. So flight initiation distance on the y-axis and the distance from across the boundary, the x-axis, with the boundary being the dotted line. And this is from the control areas, as I've said, and we found no changes in flight initiation distance across any of the 3 fish borders for any of the 3 families. So there's nothing going on in the control areas. Moving on to the reserve areas. So again, the different coloured dots are different reserves, and this is just the data for the Catchability. There was no significant increase again in FID as you move from 200 meters inside to 200 meters outside of the boundary for the non-target Catchability. But moving on to Acanthuridae, we did find significant increases in FID as you moved across the boundary for each of the reserves. And if, just as an illustration, this is the maximum mean effective range of spear guns. We didn't actually measure this in the Philippines, so this is just taken from Vanuatu, actually, this one in particular. And we didn't include this in our analysis, but just for illustrative purposes. You can see that inside the marine reserve, most of the fishes are below this range, and it's not until you actually get quite far out of the marine reserve, around 100 meters, that you start to see the majority of fishes will be beyond this range. The Scouridae showed a very sort of similar pattern, where you had a significant increase in FID as you went from 200 meters inside to 200 meters outside the marine reserves. Again, we saw this at all 3 marine reserves. Scouridae tend to be just generally more wary than Acanthuridae, so when you look at the mean effective range of spear guns, it's not quite as pronounced, but again, you can see that it's not until you get quite far outside the marine reserve. The vast majority of FIDs that were estimated were beyond the mean effective range of spear guns. So basically, I hope you can see that FID does definitely low FID does extend beyond the boundaries of the marine reserve. So we now wanted to compare this to biomass spillover. So in the way I did this is I created models using the non-linear models using the data from all collected across the 3 marine reserves to create a model FID as we went from inside to outside the boundary. So that's what these blue lines are. The dotted lines around them are the 95% confidence intervals for the model. And these sort of grey regions are the mean FID in fish control area. So what I'm using to basically define how far biomass is I'm saying that spillover is that I'm saying that we have an effect of the reserve until FID starts to exceed the mean of the fish control areas, and that's where we can say that the sort of reserve effect stops. And so these grey areas are just a 95% confidence intervals around that intersection. And what we found for both Acanthuridae and Scaridae is that FID, so this decreased FID extended to about 140 metres outside of the reserve border. When I looked at the biomass data that I collected from 200 metres inside to 200 metres outside and modelled this using the general logistic decay that's being used in all of spillover modelling, what I found is that when you look at, again, the mean biomass in the fish control areas, I'm using the same sort of indicator for reserve effect, is that we found that the intersection between the model and the mean biomass in the control areas was at or just inside the border of the reserve. We also found that the fish biomass is lower immediately outside of the reserve, so that not only is not really an effect of spillover that we have detected, what we're seeing is that just outside the reserve you have a much lower biomass than you would have in the control areas. And so the sort of conclusions that we can gain from this is that this FID effect extended approximately 150 metres further than we saw a reserve effect in biomass, and this is a previously undocumented benefit from marine reserves. People hadn't previously shown that fishes expelling out of marine reserves could potentially be more catchable. But the sort of flip side of this is that the higher catchability that we've shown that could occur near marine reserves may actually lead to local depletion quite close to the reserve. And this means that catch data that is widely used to inform fisheries management may not reflect abundance. So these areas possibly still had a very high catch per unit effort, but this probably wasn't reflective of the abundance of fishes there, and possibly catchability was what was driving it. So just my final conclusions from my work and the further questions. First of all, fish behavioural change is an important effect of fishing management, as I hope I've shown today. It provides an additional and tractable benefit to local fishes, both through spillover from permanent marine protected areas and also through harvesting of temporary periodic closures. And vulnerability to fishing that is generally assumed to be constant across small spatial scales may not actually be constant within that spatial scale. We may have this change in vulnerability to fishing, which then has impacts on catch per unit effort and what we use as indexes of abundance. Further questions is where I see this research going, or things I would like to look at. These are only a few of them. I've got 101 questions at the moment. The first is what would be the community effect of optimizing harvest for FID benefits. So we saw from my Vanuatu work that FID has basically rebounded and come down low after 6 months of closing. And then it was lost in 3 days. But we don't know how long it would then take to come down. We just know that it has this sort of like 6 months, that after 6 months it's come down again to make fish much more catchable. But if we optimize it so that we take the most advantage of the FID, what then are the effects on biomass? Will we then see strong fishing down of the standing stock? What are the implications for ecosystem function? Again, from Vanuatu, we seem to be removing Acanthuridae preferentially over Scaridae. And if we have sort of reduced catchability and we're removing these some important functional groups, then could there be any sort of like knock-on effects to ecosystem function caused by periodically harvested closures or in areas close to marine protected areas? What are the mechanisms behind this? We don't know how fish are transferring the sort of information. There's strong sort of evidence from various other terrestrial work and from sort of like stream-based studies that there could be a genetic component to wariness. And so in places like Chagos, where there's no fishing, it could be a genetic component that's got genetic influence going on. Whereas in places like Papua New Guinea and Philippines, where there is quite a lot of fishing, you may just have a generally a more wary pool to draw from. But the other thing that could be going on is that you just have transfer of information between fishes that have experienced fishing, whether it's through olfactory mechanisms or whether it's through social learning. We're not sure. So I think this is probably going to be a fairly big field to study. And finally, one thing that I'm particularly interested in looking at is, does FID have a role in reserve monitoring, particularly in compliance with reserve monitoring? I think that FID is a fairly sensitive tool, and it's a relatively easy tool to teach people to use. So it's quite easy and quite quick that we can get the results from. And so looking at things like compliance, this might be a way that we can look at compliance. But we do need to do a lot more work in looking at the different factors that revolve around FID in terms of whether or not it can be used for this. Finally, publications from my PhD. So the spillover chapter was published in Ecology Letters. The first work I did in Papua New Guinea was in PLOS 1, and then the sort of methods paper has been published in Marine Ecology Progress Series. I've currently got the Vanuatu stuff in review at Aquatic Conservation, and I've got a couple of other papers in the pipeline that I'm looking to prepare and submit. I've been fortunate to be involved in a couple of other publications, and particularly Josh's one in PNAS last year, which was basically stemmed from the work I did in Papua New Guinea as well. And I've given presentations about this work both at ICRS in Cairns last year, but also at the American Fisheries Society and also at the University of Wisconsin-Madison. And finally, acknowledgements. I've been helped by a lot of people during this work. I'd particularly like to thank Rebecca Lawton and Kirsty Nash, neither of whom are here today, for all the assistance and fieldwork in Vanuatu. Same with Rene Abisamis for his help in the Philippines. And also Aaron McNeil and Jonathan Cool have helped me a lot with some of the statistical stuff that I've had to work on. Funding was generally through the ARC Centre for Coral Reef Studies, but there's also some funding from the Josh's National Geographic grant, and I also received some funding from the JCU Graduate Research Scheme, Wildlife Conservation Society, Apo Island, various communities that I've worked with, and I particularly want to thank Steph Januchowski-Hartley, who I met and married 6 months after starting my PhD.
All right, we'll take a few questions.
On that last comment, I wondered who was the predator and who was the prey.
You're obviously after them. Any questions?
Are there any evidences of this happening with books and not squid?
So one of the things that might be happening, and something I need to look at from the broader scale country, is that spear gun fishing is of differing importance in each of these countries, but other fishing gears are of importance. So, for example, in Papua New Guinea, basically it's split about half and half between spear fishing and line fishing, whereas in Vanuatu it's totally spear fishing. And yet we still found an impact of FID. I know that Dave Feary is doing some work on this at the moment. My personal belief is that, yes, we will find a difference, because FID is just a measure of fish wariness and fish aggressiveness. So, and fish wariness and aggressiveness has been shown to influence whether or not fish will take the hook. So this could be used, yes, to look at the effects of line fishing.
Yeah, Adrian, he did look in Sydney. He's looked at it. It's really interesting there because there's areas where you can line fish, but not spearfish, and there's other areas you can spearfish but not line fish, and there's open areas and there's reserves. So he's looked at all those treatments, and he did find an effect in the areas where you can line fish only, but it's not as strong as where you can spearfish.
Yeah, I haven't seen that work yet. We'll see it hopefully.
Fraser, did you get a sense anywhere with the spillover of the decreased FID distance? Was it related to reserve size at all?
Well, all of these reserves were actually really small reserves in the Philippines. They're sort of like 400 to 500 meters of reef front. So I couldn't really say whether this related to reserve size or not. It may well be that as you move further into reserve, for a big reserve, you might actually see FID decrease even further. The distance beyond the reserve, whether that will change, I'm not sure.
Great talk. I guess the question that I had sort of throughout your whole talk, and so I'm not sure exactly which component it fits with best, but you nailed it when you talked about mechanisms and your future direction. Would you expect to see the same trend in a long-lived species versus a short-lived?
Well, I think some of the species that I was looking at do live for 15 to 20 years, so you know, we're definitely seeing this trend in long-lived species. As far as short-lived species are concerned, we're getting increase in flight initiation distance in even young fish. So, small fish, which I assume are younger fish. So there may well be an effect on sort of like short-lived species, but I wouldn't actually like nail my colours to not what that effect would be.
Do you think there could be any effect of an increased number of non-human predators in reserves? I mean, it looks from your data that maybe there would be, but do you have any thoughts on that?
Well, a lot of the reserves that I was studying were in very heavily fished systems, and they're quite small reserves, so there aren't actually many large predators within the reserve, especially not large predators in the fish that I'm looking at. So there's a general sort of theory that the maximum size of a predator will comfortably eat, which is about, basically its body depth is like 0.6 gape size. And so for a lot of these fishes, which are 25-30 centimeters long that I'm looking at, they've effectively escaped a lot of the predation that's happening on the reef. In places like Palmyra, where there's a lot of sharks, or in Chagos, where there's a lot of sharks, there could possibly be an effect, but from the small amount of analysis done in Chagos data, there doesn't seem to be. It seems to be that human predation on sort of like the size scale that I'm looking at is what's really driving stuff.
I think I'm maybe not as stealthy as a shark. I didn't seem too worried about me until really soon.
Fraser, what would you expect on reefs where you have scuba diving only or tourism activities and you don't have any fish? Do you think that the fish would react in a similar way as they would in areas that were closed because they saw us not necessarily a threat?
If we're talking like in a very large system that's been closed from fishing, I'd expect there to be you'd see a much lower FID than these sort of like small areas. The studies that have been done there's been a couple of studies done looking at scuba divers and sort of like how fish are getting attracted to them, and it's generally through scuba divers basically stirring up the sediment or breaking coral or feeding fish. It's basically about this sort of like they're making food more available. So in general, I think that yes, if an entire big area was closed off, we might see a bigger effect than we currently are, and there might be this impact of the fish situation, this sort of idea that fish might come closer to scuba divers just because food might become more available.
Sorry, I guess slightly related to that. One of the things that came across to me earlier that affected flight initiation distance was the resource availability.
Yes.
And I was wondering what reflections you had on that or what you noticed.
So I tried to control, but it was actually particularly difficult to control for in terms of like the small sites, so like the sort of small patches that fishes were using. I did do benthic surveys across every area that I looked at, and I was looking at things like turf algae and coral cover and so on and so forth. In general, I found that there was no real influence of whether looking at things like macroalgal cover, turf cover and so on. There wasn't really any influence of that in FID.
Right, way back in chapter 1, you mentioned the presence and absence of a spear gun in the hand. And I got a bit confused because you said I couldn't do that in reserves. Did you actually do it somewhere? The presence and absence?
Yeah, so one of the reserves in Vanuatu, because we were doing all of our work with the reserve chairman, the guy who basically was overlooking all of he was the committee chairman, he was overlooking all of the reserves. He had because we had him in the boat and so on, us doing that was more socially acceptable. But in places in Papua New Guinea where they spent a lot of time, a lot of effort, basically trying to get these reserves in place, and there's sometimes quite a lot of friction in the village around them, taking anything that looks like a spear gun to reserve, even if you're not coming up with fish, is probably considerably less.
And your conclusion seemed to be that it didn't make any difference.
No, it didn't seem to make any difference. It seems that fish are actually taking their cue. There's a bit of literature that looks at where fish take their cue from, and it's saying sort of like distance between eyes and mouths, and sort of like room size, and basically the eyes of the predator. So it seems like they're much more taking their sort of like cue from your face rather than.
That's a really surprising result, because I mean, a spear gun is increasing your distance to. If you put the spear gun down, the fish will behave normally. You pick it up, and they detect it straight away.
Yeah, so one thing that I did find, but I wasn't sure how that I could really attack this in the analysis of it, was that if you move a spear gun, if you're basically attacking the fish in a straight line, the spear gun held in front of you, you get this sort of significant difference. If you're within a certain distance of the fish and you move the spear gun, whether it's from pointing at it to point at it or in any other way, you'll see the fishes respond to that. So that's one of the reasons why I started all of my approaches from well outside what would be flight initiation distance.
Yeah, there's so much speed fish most of my life. It seems like a strange conclusion, but having said that, you've standardised the methodology right across, so there's no issue.
It seems to be movement within across planes within a certain distance. But again, this is something that that's like one of my 101 questions. Is that because I've talked to a lot of spear fishermen and they say, yeah, you basically can't move this. Basically, all said you can't really move spear. It's very when you're close to a fish because the fish will react. You need to be pointing at the fish from distance. Great talk.
Okay, I think we'll wrap it up then. Thanks, Fraser, one more time.